So yeah, we're doing actually the third fruit fly experiment in the last 7-8 months. We had one on SpaceX 3, one on 4, and now one on 5. And so let me start out by telling you a little bit what this experiment is about and what's unique about it. And then I will go on to giving you a little bit of a background of what the other ones were so that you get the big picture of why we're interested in using fruit flies in space. So one question that a lot of people come up with is like, fruit flies? Really? You know, what's the excitement about fruit flies with doing science? Now, something that you would have heard from a lot of the speakers earlier is that the big picture is that we're interested in humans being in space for a period of time. But you'll know that there's a lot of experiments you can't do with humans. You can't do a lot of the genetic stuff. You can't do dissections. You can't extract, you know, tissue samples and look at the molecular biology of what happened. You can extract some from humans, like blood samples and urine samples, but there's a lot that you can't do for the human, and yet you want this information for the human. So fruit flies serve and have actually been used for the last over 100 years in biology labs across the world to understand a lot of very fundamental things like, how does your heart function? How does your immune system function? How does aging happen? How can you reduce aging? All of these are questions that you can actually answer, and many more that you can answer with a fruit fly. So what we're doing on this mission on SpaceX 5 is we are flying, so I'm actually very excited because the last 15 years that I've been working at NASA, this is what I've been, you know, hoping to get to the public, which is a facility that scientists all over the world can use to do Drosophila experiments in space on the International Space Station. So what's unique about this particular hardware, this facility that we're putting up, it'll have the capability to, actually many things. So one, it's going to have the capability to have what is called a 1G control. So you have a centrifuge so that half your samples, actually a third of your samples are in the microgravity environment of space. A third of your samples are in a 1G control in space. And a third of the samples then are on the ground as your control. So the advantage of this is if you see a change in space flight, you can now start asking the question of, was it the micro-G environment that did it? Or was it radiation, vibration, something else from space flight? So having the two control groups is a really nice way to do the experiment. So that capability is available on this facility. There's a high-definition video camera, so you can look at the status of the flies, the health, how many are coming out, you know, when are the children and the grandchildren being born, you know, for the flies, and monitoring exactly how long development took. You can study how development is happening, because the fruit fly actually serves as a very universally accepted model organism for studying development. And that same development, many of those same processes happen in human development as well. So there's the camera, there's lighting. So just like on Earth, we have 12 hours of day, 12 hours of night. We have the capability for circadian lighting in this hardware. We also are, for the first time, going to take samples and do fixation with them. So scientists who want to fix their samples will have the ability to do that. What else? Oh yes, of course, multi-generational capability. So we can now, as many of the cassettes that we send up, and I actually really wanted to bring a cassette for you, but you'll find pictures of it all over the web because we have a lot of outreach things that we've done before. So just search for my name, Sharmila Bhattacharya, NASA Ames, and you'll see pictures of our hardware. But we can actually separate generations, do multiple generations, and do studies that way. So that's the facility aspect of this experiment that's exciting, because this is now a capability that anyone can use. You write grants, you use it through cases, you use it through NASA, you can do it through foreign entities applying through their space agencies as well. So there's that. And then the second part, that I'm a scientist, so the part that excites me also, is the fact that we're also going to do an experiment here where we're going to look at the immune changes. So we're going to look at how well does is the fly able to fight an infection, just like you fight off your cold or a stomach bug or something. So we're giving the fruit fly an infection, something that's pathogenic to the fly, and we're going to watch how effectively it's able to counteract that infection in space. So that's the science aspect of it. And then we'll go to questions, but before that, I just wanted to wrap up with what were the two other experiments that I just talked about that many of you heard and were here and we discussed earlier for SpaceX 3 and 4. We've started getting some very exciting results on some of those, and just to give you a little idea of what else you can do with these small critters that you most likely want to swat in your kitchen or in your bathroom, but that serve as a very useful tool to scientists. We've been using, for SpaceX 3, we used it for a cardiovascular experiment, and we're getting some very neat data on flies that developed completely in space, and how the spaceflight environment altered cardiovascular function. And many of those functions actually mirror some of the changes that you see in astronauts. Same with the earlier experiment we'd done before that on the immune system. Many of the changes that we saw in the immune system also mirror what you see in humans. So in other words, they are very useful tools to then understand the molecular biology of what's happening in the body, because instead of now having 6 astronauts in space, all of whom are of different ages, different sex, they are from different ethnic groups, therefore different genetic backgrounds, they eat different food, they have different predispositions to anything that you can think of. Here we have, in a box this big, 3,000 animals that are identical, or 3,000 flies that are identical, that you then bring down and you do your analysis. So there's a lot of statistical power to using these simple models and then understanding something in a more complex system like the human. So I think with that, I will stop and take questions. Yes?
The rest of the multi-generational aspect of having flies,
yes.
What big questions are you asking as far as like physiological, genetic adaptations, or period of time?
Excellent question. So we did ask that question for the cardiovascular experiment, for example. And there is actually what we're seeing, and we haven't published this data yet, so maybe in the next year or year and a half, we're hoping to get this out. But there are distinctly adaptations that happen with time. So just to give you an example, and this part I can tell you from samples that come back to the ground, the different amounts of time that they spend on 1G after they come back from space, the adaptation, depending on how long they were in space, how long they took to adapt back to 1G was dependent on how long they were in space, for example. So yeah, adaptation is definitely something we're looking at, and how long they spend in space definitely is something that affects the biology. And what we want to see with multi-generation is, I think, exactly what you're getting at, which is with many and with fruit flies, since one generation takes 10 days, you can imagine that in a month you can look at 3 generations. In 6 months you're looking at 18 generations, 15 to 18 generations. So you can answer that kind of question much more easily with something like the fly that quickly turns around, where you say, okay, so you saw these cardiovascular changes in the first generation. What about the 18th generation fly that's been there? Are they better adapted, and do they show less of those problems? And those are exactly the reasons why we want to have this capability available for everyone. Yes?
I know last week we had like a certain set were male and a certain set were female.
Yes.
Is it the same? What are the numbers?
Yeah. So this time what we're sending up is very similar numbers to start the culture. So we have roughly 10 females, 5 males for each of 12 cassettes that we're sending up. But then we're going to do 5 sets of food change out. And then, so by the end of it, we get, 12 multiplied by 5. So at least 60 cassettes. And then we also have some that we're fixing. So we'll have a large number of cassettes then coming back, and those later ones will all be males and females mixed. But we'll do our analysis most likely with males and females separated, because one interesting thing we've seen from our past data is that males and females actually respond very differently in spaceflight in terms of molecular biology and physiology. So we're very interested in that. Yes.
Thank you. That's exactly what I was going to ask you. So you
Yes.
send up 10 girls and 5 boys.
Yes.
Do those in any way when they're up in space kind of statistically lead to different distribution of male, female compared to control who's back on the ground? Or
Yeah, you know, that's
similar?
a great question. So far that we've seen in our previous experiments, the distribution of male to male stays the same. So we still have the 50-50. But what changes is their physiological response. So how they respond to stress seems to be different. And it makes sense physiologically because the female fruit flies, you know, they have the burden of the reproduction, which is a big physiological draw on the body. And so you can imagine that, you know, the proteins needed to respond to stress then respond differently for the female flies compared to the male flies.
And one quick follow-up. So
Yeah.
when going up to space,
Mhm.
is there any kind of, you say there's a differentiation. Are the ones that go up to space like hardier because of all the hardship they've been able to endure and come back, or are they weaker?
Now, this is an excellent question. So there's a whole group of scientists that study this sort of thing. That is, and on Earth even, people who study, let's say if we stress out flies with, say, heat shock, does it make them more resilient to a subsequent heat shock or starvation or some other stressor? And generally, it looks like that is, that is a phenomenon that does happen to actually most animals, most multicellular creatures, which is that they do get hardier. Now, having said that, there are other aspects. So let's say when we looked at the immune system, right, for one of the experiments that we had looked at earlier, flies coming back from space had an altered immune response. So a lot of their immune genes were upregulated, meaning they were expressing even when they weren't infected. So you could argue that therefore when they came back to Earth, if you infected them, you know, they'd be able to withstand it. So that does happen in some cases. On the other hand, if you look at other systems, like if you look at, say, the bone or the muscle of any animal coming back, those do get weaker. So it depends on what system you're looking at. Some systems are weakened and others are not, are not compensated. So that's a great question. But in general, therefore, spaceflight is a stressor which definitely alters a lot of physiological processes, and very often differently, and differently between males and females, differently from one tissue to another, differently depending on the age. The older animals respond differently from the younger ones. So, yeah. Yes.
Can I follow up his
Sure.
awesome question with a silly question?
No, no question is silly. It's all good.
How do we get around? How do they bounce into the wall?
That's actually an excellent question. And you know why that's an excellent question is, and really there is no silly question, so please ask anything that comes to your mind, including how do they poop, which everyone asks the astronauts. So the reason the behavior question is actually a very good one is our last mission, SpaceX-4, the one we flew, was actually looking at exactly that, which is that Drosophila flies, which means that we are very sensitive to gravity. All organisms that evolve on Earth are. But flies, you can actually even look at it and assess it better because they fly, which means that they have to always know where the gravity vector is, what the direction of the gravity vector is, what are the air currents happening. So they're very acutely sensitive to the gravity issue, just like birds and so on, because they're flying entities. And so one of the questions we had is how do the flies, and do they adapt? The question that she asked earlier about adaptation came in, is something you can assess easily with behavior. For example, if they're tumbling and behaving oddly at the beginning, but by the third generation, now they're flying fine, then that's an adaptation that you can actually measure, and you can say, okay, how long did this take to happen? So it isn't at all a bad question. So in terms of behavior, I can tell you a few things. We've looked at the behavior videos now. We haven't had time yet to do all of the mathematical analysis of what it means, but qualitatively looking at the videos, flies are able to fly, which didn't surprise us because we know that they mate, because on previous missions where we didn't have a camera, we still knew that the next generation developed. So they're able to find each other even when you have a few animals. Like when you send up 15 in a habitat, they have to find each other. By the end of it, you have 100 in the habitat, and then you kind of bounce off of each other, so it's much easier. But so they are able to move. The thing we want to look at is, are they more likely to walk than fly? Because when they're walking, they're using the wall as support, right? And when they're flying, you would think that the gravity factor becomes more important. So those are the sorts of things we want to see. And then also when they fly, do they fly normally? Is their path and trajectory normal? Or, do they tumble? Experiments that were done in the past with bees, which are bigger, showed that the bees were tumbling. So it's almost like they were looking for the gravity vector. And what would have been interesting is to see, what happens if the bees then were allowed to be there for 10 generations? Do they then adapt? Are they better off? But again, those take having multi-generational hardware and doing long-term studies. And so what I'm hoping on SpaceX-5 is this is the beginning of a facility that will allow studies exactly of the kinds that you guys are asking questions of.